Please come in the front. So, welcome everybody. Close. Okay. So, welcome everybody to this GAN course. It's not working.
No, not that, not that.
No, it's hello. I have to take very close.
Yeah.
Hello.
Yeah.
So, welcome to this GAN course, which is titled Physics of Living Systems. And we have with us Professor Daniel Revelin from France. I will introduce him in a minute. So, he'll be with us for next 5 days, and you will have opportunity to interact with him. So, I welcome all the participants. So we got a good response actually. About 50 participants registered for the course, but maybe because it is online also, so we shared the link. I don't know if that has led to some kind of EA. So we'll try to make sure that more and more students attend over the next 5 days. Actually, we have been planning for this course long time back. Before COVID, we started discussing about the course, but due to COVID, actually, things got halted, and Daniel was pushing for this course. He was really interested to come JNU and teach this course. So then when the GAN call was open, we actually applied for the course, and then it got sanctioned. So we are very happy that Professor Daniel Revelin could be here with us. So without taking any time, I would like to invite Professor Daniel. Please take a seat. It's a formal 5-minute introduction. And also, I would like to call upon our Dean, Professor Shobhan Sen. Please take a seat. And our coordinator, GAN coordinator, Professor Satyal Patnaik, also. So as per the registration, we have participants from different universities like Ashoka University, Shivnadar University. Some of the students are from School of Biotechnology in JNU, Life Sciences. So I was hoping that the students, they are from wider backgrounds. They have biology, chemistry, and physics kind of background. So hopefully they will enjoy this course. Uh, Also, I would like participants to take this opportunity actually to interact with our speaker and learn more and more about the topic. Okay, so this is a good platform. I invite now Professor Shobhan Sen. Please, because many of the participants are new, so please tell us something about the school.
Yeah. Welcome you all in this, I think the last GAN course. Is it, Satyabrat? I think the last GAN course, which will be completing the 12th course in running in JNU, where we have Professor Daniel Revelin from Strasbourg. And it is quite nice to have him finally, because we were thinking for a long period of time. I think Satyabrat, myself, and Sanjay Puri were visiting in Strasbourg, I think 2019, is it? And then at that time we heard his lecture, and we were so impressed from his lecture. And we thought, no, you should come to our department and have some, lengthy course of this. And then we continued interest, and we are happy to have him finally over here for the next 5 days. Regarding the School of Physical Sciences, I have the great opportunity to welcome you all on behalf of the School of Physical Sciences. As you all know, this school is a hybrid of all the 3 natural science subjects like physics, chemistry, and maths. And that's where we should pitch in. In fact, you can see that it's all about the physics of living systems. That means you are combining your physics ideas into biological systems, and that's exactly what Daniel is going to show us. How you actually can apply physics models to really do some kind of a biological system to be understood in terms of the development biology and many other things. He's a great scientist in many ways. I think Manoj will probably do a more formal introduction about him. Anyway, so without further any delay, so let's have this program and the course running as smooth as possible. Also, this course will have the hands-on part of it. In fact, I think this is one of the courses in Gyan that we'll have also, before lunchtime we have the lectures, and after lunchtime we have the hands-on training. That will be happening in our master's chemistry lab, where we are going to show some simple, normal, simple microscopes you can actually use to do this kind of simple analysis of biological systems. So we have some plans. We were doing this planning for quite some time over online meetings and so on. And Daniel will also show you some very interesting microscopes that he brought from France as well. So with this, let's welcome you all. I welcome you all, and let's start this process. Yeah.
I would like to Satyajit to say some few words about the GAN course.
GAN course, yes.
So good morning, namaste, and so there's never a last course on GAN. I mean, this is for this financial year. Yeah, so we hope that the advertisement will come again. So GAN, the full form is Global Initiative for Academic Network, and it is not just a series of lectures. It is some kind of network building. So some expert is here, Guru is here, and new people are here. So there has to be interaction, and possibly the next steps like writing a common project or getting a postdoc or PhD, exchange visits. So this is the idea that more nucleation of some research in a particular field. That's what the aspiration of the GAN program. So GAN program in JNU started around 2015, and this should be somewhat around 55 or 56th course. So we have been running these courses. Last financial year, we had 12 courses, and this happens to be the last one. And in some area which is overlapping in biology, chemistry, physics, and a little bit of maths, right? I'm reminded of a joke by Professor Rajaram. He told us that about what's the difference in approach between a biologist and a physicist and a mathematician. And the joke is that three scientists, one biologist, one mathematician, and a physicist landed up in a foreign country, okay? And they came out of the aeroplane and they saw a four-legged animal looking like a black color and from a distance. And the biologist exclaimed, "Aha, this country, I conclude, has black goats." Physicist was a little more circumspect and he was more kind of reasonable. He said, "No, based on the evidence available, you can only say this country has at least one black goat. You can't generalize." The mathematician is stricter, more exact. He said, "No, you cannot conclude that. You can only conclude that this country has at least one goat whose one side is black." So mathematician is one extreme, too exact. Biologists have the tendency of making it too general, and physicists probably somewhere in between and try to middle path. Is that what they follow? But we have a mixture of all three, and some chemistry also. So I think the core of SPS is interdisciplinarity. Physicists, chemists, mathematicians staying under one roof. And so I think this is a course most apt for SPS. And we're very happy that Professor Daniel has kindly agreed to our request and to initiate this program. And we're really grateful that he could make it to JNU. And it takes time to get into, it's a humanities-centric university. Social science dominate over physical sciences courses, but still there are pros and cons of JNU, and so I think everyone should enjoy and enrich. So remember, this is not just a course on living systems, physics of living system. This is a networking phenomena. Try to interact with the visiting faculty, and if possible, try to nucleate a program in this field over the next years. Thank you so much.
Thank you, Professor Patnaik. So, without taking any time, let me introduce Professor Daniel Rivlin, who has done his PhD in 1997 from Institut Curie in Paris, France, and then he took up a postdoctoral position at the Weizmann Institute of Science, Israel. After that, in 1999, he started his own group at the University of Grenoble France, right? And he established his group in the area of studying mechanosensings of contacts with extracellular matrix and between cells. Okay. So in 2010, he moved to Rockefeller University for sabbatical, and after that, after finishing his sabbatical, he moved to Institute of Genetics and Molecular and Cellular Biology in 2015 in Strasbourg, where he continued his research in the area of self-organization living matters. So currently he is a research director at the French National Center for Scientific Research at the University of Strasbourg, France. So with this, he has lots of publications. You should visit his website. He has very highly impact factor rich publications in Nature, biology and others, Nature Physics. So please visit his website, and I would ask participants, try and interact, because as Professor Shobhan Sen said, we have a experimental hands-on session in the afternoon where he's very excited about these experiments. So he's going to hopefully keep you engaged for the next 5 days. So try to interact. He's going to show some biological phenomena with the microscopes. We'll ask you to handle the microscope to see the matters moving around. So it will be very interesting. So with this, I would like to invite Professor Daniel to start his talk.
Okay, you can start.
All right.
It's okay. Yeah, come on. Just take it out.
If I speak like this, can you hear me? Or you prefer to the microphone?
You need to put your microphone.
I need to put my microphone.
No, their microphone. This one is better.
Okay, sorry.
No, it's not reader. It's not reader. No.
Some students will be following you through online.
Yes, so I should have.
They'll also ask questions online.
Okay. And somebody will tell the questions? Somebody
So
will
yeah, they will.
tell the
There is
questions.
someone
Okay.
else.
Right.
If somebody is.
Could you ask the students to come closer?
Students, please come closer. That's what you know.
Yeah, the closer you are, the better. There is a lot of room here. Yeah, no, close. Yeah, sure. First row, second row. Yeah, sure. Come. Yeah. Come. No worry. It will make the interaction easier.
You put it inside the pocket, no problem.
Inside the pocket, that should be all right. Okay, you sure? You come? No? Okay, maybe later on during the week it will come, but I think it will make the interaction easier. First of all, I would like to thank Manoj, Shobhan, etc. It's a pleasure to be here. Real pleasure for many reasons. I arrived yesterday evening with a wonderful welcoming, and also it's a pleasure to see you, new faces. For me, I hope we'll get a chance to discuss this week. As you will see, I don't want to use PowerPoint too much. The idea is to interact with you and to try to communicate my passion on biological physics. That's really the intention. Not about, this particular university, NGU, being the humanities. Historically, most of the very original features were discovered by the humanities in the 18th century or the 19th century. Philosophers were speaking and thinking about science in a very clear way, and eventually they got to key discoveries, and this gave many names of many equations, many phenomena. So you can consider yourself in a place where you could be yourself very creative and make breakthrough in science also. So just, that's, I think, one way to see it, which is historically proven and quite true. So today, I'll be giving you an overview about what we're going to do during the week. I will ask you many questions, so please speak up, right? The idea is that you shouldn't be shy. It's a lecture, it's not a seminar, so the more you speak, the better. And one of the ideas, I understood that most of you are coming from chemistry. Could you raise your hand, the ones who come from chemistry? Right, good. So I was when I came back from sabbatical, I was in Supplementary Chemistry Institute, so I was surrounded by chemists. Physics? Biology? Wow. So a true mathematician? No? Engineer? Right. Good. So basically, the way I would like to do is that I will ask you again many questions, and please speak up. There will be some equations, some biology, to some extent some chemistry, but I would like to keep it at a level so that everybody is on the same page. The structure of the lecture goes like this. This morning it will be really an introductory lecture about biological physics. This afternoon there will be hands-on, as was introduced before, and every day we'll have this format about lecture in the morning, the afternoon experiments, and we'll run through this until Friday. And on Friday, returning to the idea of the humanities, we'll go through an old article very slowly and trying to think that the old articles sometimes have really deep understanding and deep vision about what the science is about. So that's the plan of the whole week. Right. At which scale is the biological information conveyed? At which scale is the biological information conveyed?
Length or time scale.
What is spatial style? Spatial scale. What is the spatial scale that is the most relevant that you think about when you think about biology? Yes, speak up, but one after the other. Right. So
The microscale to nanoscale.
nanoscale. You would say that the nanoscale is the right one. So basically, I would like that we go together through a scheme that will go through this, which is going to be the spatial scale. And we're going to build very slowly some arguments about, scales, and essentially scale. So you said nanometer, if I heard correctly. So I totally agree with you. At the nanometer scale, what would you place? As a key, key biological information that is essential to convey life or to make the living matter be living matter.
Size of cell.
Any molecule that would, be relevant to convey biological information. I'm sorry? DNA, that's right. What else?
Molecular proteins might have.
Proteins, that's right. Other key objects that somehow you would find in any living matter that you would find everywhere. Protein, DNA.
The lipids also.
Lipids, that's right. What else? What do you say? Carbohydrates, sure, yes. I mean, there is between DNA and protein, you forgot also one element that we talk about, RNA, sure, RNA, sure. Right. And basically, I'm saying that this is conveying the information because if you want to make a distinction between inert matter and living matter, most of the time, or even all the time, you will find a key difference because you will find DNA, RNA, protein, or lipid in this living matter, right? So, and this is at the nanometer scale. Now, if you think about the cell, what is the scale of a cell, a typical cell? Micrometer, right? So I'm spanning scale, and I'm going to go at the scale of the micrometer. And if I'm talking about the micrometer, could you give me an example of a cell that is 1 micrometer in size? What do you say?
Bacteria.
Bacteria, that's right. So we write bacteria. Could you give me a scale of our cells? What is the typical scale?
10 microns.
10 micron? Everyone agrees? 10?
Every time, but like the average will be.
Yes, 10?
It goes beyond that also.
Beyond that? Okay, right?
Maybe like 5 to 50.
5 to 50. So there is a range, all right? So let's go like 10 micrometer. I will say collectively mammalian cell. All right. What is the difference between bacteria and mammalian cell? How would you qualify the key difference between both of them?
The arrangement of nucleic acid.
You need to raise your hand because otherwise, yes.
The arrangement of nucleic acid inside the cell.
Fair enough. That's one difference. There is another one that is canonical. That is, yes, I'm hearing many men and not many women, if I may say. So, and I know they want to speak. So please, women, speak up. Yes, you need to shout because I don't hear. Yeah. You know, if you come close, cell wall, that's right, that's fair enough. Cell wall is one difference. Another key difference that is in the nucleus. What do you say?
Loud.
Nuclear membrane, presence of nuclear membrane.
Nuclear, that's right. So basically, the prokaryotes have essentially no wall inside the nucleus, where eukaryotic cells do have it. If you change your mind and come closer, that will make easier the exchange. So please come. So these are the prokaryotic cell, and these are the eukaryotic cell. And essentially, the difference is going to be about the fact that there is a compartment for the nucleus, and there is no compartment for the nucleus. You see that here there is 3 orders of magnitude, right? From nanometer to micrometer. This is gigantic. It's 10 to the 3. So there is something that is already very, very surprising in the fact that if you think about a cell, either a bacteria or a mammalian cell, you span the information that is meaningful, the genome, from nanometer scale to the micrometer scale to 10 to the 3. And it's working pretty fine. So there is something special that is at the core of biological physics. Somehow. How are you going to have 3 orders of magnitude out, which is going to be working? To give you a metaphor, imagine this building, right? There would be a brick in this building, which would be the nanometer object, and I would take 10 to the 6 of these bricks, I will throw them like this, and I will be hoping for the best that this building comes about. The probability to have a building that would be sustainable is minimal. Yeah, I think you will agree with me, no problem. So there is a problem of change of scale if you go from this scale to that scale, which is a real problem, and the system has to be pretty robust at the same time. Now, tissues. What is the scale of a tissue?
So they're really, really big.
Millimeters.
So tissues are really big compared to.
Big. So you need to give a scale, and please raise your hand, and the one that please, yes. And then the one that I didn't hear, speak up, yes.
So if we compare, skin is also a tissue.
Yes.
Yeah, really high skin. And then maybe gut is also a tissue.
All right, so basically you will give me which scale? What would be a typical...
Sir, we should go maybe a few millimeters also.
You go to a few millimeters, fair enough. So I will go to millimeter. I could go also to 100 micrometer, and I propose to draw epithelial cell that we'll talk about also later on this week, like this type of structure, a honeycomb, where each of these objects is a cell, right? And as a whole, I'm going to form a tissue which is going to be an epithelial layer, an epithelial structure. Now again, I'm changing scale. I'm going to 10 micrometer up to 1 millimeter or 10 or 100 micrometer. I'm turning that epithelium because you would find this type of epithelium inside many organs in the body. You would find them in the pancreas, in the intestine, in the gut, etc. basically. And the basic rule is that you would find this type of structure with adhesion between the cell, the nucleus, and a proliferative unit that is going to give tissue be what it is. Net scale. And I would like to ask you to raise your hand, and essentially the one that I did not hear so far. What comes after the tissue? What do you say? Organs? That's right. Could you give me different organs? Yes? Okay, so organs. So I'm going now to a millimeter, let's say 1 centimeter or like. So you said the heart. Before that, you said? Liver. Pancreas. Intestine. Etc. Right. Right? Again, I'm changing scale. How in 3 dimensions am I going to go from this to this particular scale? And last but not least, after the organs, which are the scale do I have to consider eventually? What? Organism. So the organism is going to be, let's say, it's going to be anything between centimeter to meter, and that will be the organism. And here, you can let your imagination run. Human being, Drosophila. Drosophila is what? Is a fly. So, nice. Okay. Nematode, you think about which system?
Ascaris.
Yes. Or another one that is heavily used in the laboratories, which is C. elegans. Another model system that also is heavily used among fish, zebrafish. Why is zebrafish used?
It's transparent.
It's transparent, so it's particularly useful to be used. And mouse, planarian, axolotl. You continue the list. The list is extremely large, but at the end of the day, you end up with a list of systems which are model systems because you can test hypotheses quite easily with this system. There is one that we did not mention, which is the plants, which also would enter into this category. And with significant difference, but nevertheless, you end up with this type of structure. So the point that I would like to highlight is that we've been spanning from the nanometer to meter here, which is a huge gap, right? We are 10 to the 9 in terms of scale, and this is quite extraordinary that you manage to go from this meaningful information up to the cell, to an ensemble of cells, to the organ, and eventually to the homeostasis of an organism. And one of the core questions of the physics of living systems somehow is to try to contribute in the understanding of this change of scale. And one of the key elements that we'll be talking about a lot this week is the concept of self-organization. Can anyone give me a definition of self-organization with everyday's life, right? Just when you think about self-organization, what comes to your mind?
This one hard sheep, so hard of sheep. This one also crowd.
Crowd, right, crowd. A typical image is the good metaphor is the crowd, right? Whenever you go to a place which is heavily crowded, you don't have any individual, wish to go to the right or to go to the left, to essentially go where you can go. So you have pressure from the side, and eventually you follow the crowd. So the crowd is a very good metaphor. Now, if you think about the crowd, you wonder how this change of scale can eventually happen. Because if essentially there is a directionality in the crowd, the fact that you're going to have a potential homeostasis or the good formation of an organ is a problem, right? How is it eventually going to work? There is a key paper that we won't cover, I don't think we'll have the time to do so, which is a paper by a physicist named Anderson, famous physicist. The key word is more is different. And it's essentially this idea that somehow if you have an ensemble of objects that are interacting together, you will have a new phenomenon that has nothing to do with the addition of the individual elements. If you want to think about it, again, return to the crowd effect. That's the easy one. If you enter, I think there is a subway in New Delhi, as I understand. So if you enter the subway and there is nobody in the subway, you will select the seat, usually far away from a neighbor because you want to be alone, right? And so basically, and in rush hour, no way you can do that. So basically, the ensemble behavior will be different from the addition from the individual behavior. So one of the elements that is quite surprising when you enter into this field, or if you come from any field of chemistry, biology, or physics, actually, is, as I try to say, this change of scale that eventually is going to give a functional organ. The last element I want to give before going now to more specific about, this particular scale for today that will be useful for this afternoon and for tomorrow is the idea of cell identity or cell state. So for the biologists, I think it's quite easy to think about it, but for those of you who are not biologists, I will go to the following route. We all started by a single cell. Once upon a time, we were a single cell. For the non-biologists who are used to this kind of category, how we ended up being what we are now? What did happen? Evolution, that's one way to go, but I was born late in the evolution, so were you. So basically, how, what did happen? When we start by a single cell, what's happening next? Cell division. So you start by one cell, then you divide, then what? You divide. But if you continue dividing like this, you end up with a ball of cells. So how eventually you end up with an embryo? there is a problem, right? I mean, if you start by single cell and you keep proliferating, if nothing special happens, eventually you have a ball of cell, and that's what you see under the microscope eventually.
Differentiation.
That's right. So this differentiation is going to be a key word that is going to come, or what do you need to change inside the cell? What needs to be changing over time? Expression pattern. So somehow the gene or the genome that is here is going to have some patterns of expression that is going to change over time. If you think about it, try to build yourself an embryo, synthetic embryo, where you would start from scratch, have the cell proliferating. If you want to express some genes that are going to make a cell be the pancreas, or the cell be the intestine, or the cell be in the heart somehow, it has to be at the right place and at the right time. So the pattern of expression is going to be absolutely key. So behind the scene, in this particular part actually, there is the idea of a cell state, called also in the biology community cell identity, which is going to be essentially the patterns of expression of genes that are going to make a cell be what it is at some point. And this is absolutely essential in the self-organization concepts, actually, in all the organisms that I'm talking about at the moment. I'm mentioning this because nowadays it's possible to make some sequencing which is spatially resolved for the transcriptomics over time for most of the model systems that are here. So it's not becoming anymore an illusion that eventually this would be possible. It's now possible. So you can have the single cell sequencing during the growth of a model organism, which is allowing you somehow to make some quantitative statements about the development of any model organism. And this is now, it is happening now, and the community needs chemists, physicists, biologists, engineers, because there is a huge amount of data that is generated and very poor knowledge about how to integrate it. Since I mentioned before this paper from Anderson, in the context of cell state, I want to mention another scientist who's been writing also a key article. I don't think we'll have the time to go through it, but I want to mention him. Anyone knows who talked about cell state very early on in the 50s? A mathematician. There is no mathematician in the audience, I understood today, but you know him. Alan Turing. Okay. So basically, he has this paper in 1952, 1952, where essentially he's talking about the chemical basis of morphogenesis. And when he talks about it, essentially he's describing the concept of cell state with the chemical state, with the mechanical state, and he's explaining how eventually an organ can form about. And you've heard about the Turing pattern in the audience? No? Yes? So maybe I'll mention that in the week. I don't know if we have the time for that, but essentially he's talking essentially about chemistry in the Turing pattern. Belousov-Jabotinsky reaction, oscillating reaction for the chemists. You've heard about it. It's a reaction that is going to be cycling. Turing predicted it, essentially. And essentially, Turing is saying an organism is an incredible meeting point between pure chemical phenomena, pure mechanical phenomena, an interplay between both. And the point that I'm trying to share with you is that now we come at a time that it's possible now to probe these concepts. And this is the whole point of the biological physics field. And now it's possible. But essentially, Turing in 1953 was already having a clear vision about how this should be made. There was a question. Yes?
Are you talking about the tape copying the DNA? There's a similarity between tape copying Turing machine.
To some extent, yes, but in his original paper on 1953, he's simply saying that if we would have at any time point the chemical composition of each of the cells during development, and now it's possible. If you read Nature, Science, Cell, every single week, you have single cell sequencing data, spatial transcriptomics very often that is showing up. He's saying, I would love, as a mathematician, he says, to have these maps over time. Actually. And now it's possible, essentially. The word that you would find if you go on Google and try to look for it, it's atlas. It's like, you know, in the old time, people wanted to have the atlas of the globe. Nowadays, people want to have the atlas of the development of the species with having this spatial transcriptomic proteomics over time being expressed. Now, once you have this, you need to integrate this into the model, and then you need to make a sense through self-organization of why this organ has this particular shape, this particular function, and so on. So what you see already with this scheme actually is that there is a whole program that needs to be written where in fact you need a large culture. You don't need to be competent in all of the field, but at least you need to get a feeling of basically how to go from one to the last one. So to wrap up this part, I just wanted to say that the idea of stem cell or cell state actually is deeply associated to the idea of stem cell. The idea of differentiation that you mentioned a little bit before. And when you think about development, you can think about stem cell proliferating at some moments, changing their identity, going to another state, eventually giving one organ, eventually sometimes transdifferentiating, reverting back to another state that it used to be before, and then you undergo another process. Two last points before we go to Brownian motion. That will be the part that we'll be dealing with this afternoon. Maybe you've been hearing about the fact that also nowadays it's possible to grow in the laboratory some organs out of the petri dish. How do you call them?
How do we grow them?
How do you call them? What is their names? What do you say? 3D bioprinting, but even without that, without bioprinting, there is a way to have something coming out of the petri dish without parents.
So it's a technology induced pluripotent stem cells.
That's right, but what it gives, what is the name, collective name, which is given to this object that you can manipulate? Organoids, okay, right. So I will put them, I don't know where to put them, they aren't exactly here, but somehow they are somewhere here. For those of you who are not familiar in the audience of organoid, it's, I think it has changed really, it's a game changer, because you can grow out of the petri dish pancreas, intestine, brain. I don't know if some of you have seen this image of a brain and coming out of the brain an eye. So then you have out of a petri dish iPSL, you make the right culture, and then you end up with the brain actually with neurons, with neuronal activity essentially out of the petri dish. And last but not least, now it's possible also to have, the word should be taken with due care because it's not the word that is appropriate nowadays, but synthetic embryo. That somehow if you start with human cells, you can end up with something that is going to look like the first stages of development, returning to the point that if you start by a single stem cell and you let it go to the right step, you end up with an object that is, what? And, right. So I just want to tell you the point that there is some unifying principle of self-organization of matter that is, at the moment, we are ready, it's happening in the laboratory, and the concept of self-organization leading to organoid and different organ of the petri dish is happening now and is not very difficult to do. So the last point I want to do, so that basically you see also where I want to go actually, is that inevitably you end up with some problem of ethics when you go to the physics of biological systems, because it's so possible and it's so easy to design these organoids or organ of the petri dish. You need to think twice before you make an experiment about the consequences of what you're starting to do. I mean, doing basic science is what we are here for. I mean, just that's the idea of basic science, but also there is a necessity to think about the far-reaching consequences. So I invite you to, we'll talk about it during tea or during the lecture as much as you want, but bearing in mind that everything that I'm going to say has to be taken with due care in terms of application and in terms of the types of questions you can ask, or you should ask. Physics of living matter, it's certainly a matter with new phenomena, but at the same time, there are some far-reaching consequences about the next steps of humanity. So even though these rules are fascinating, they should be taken with due care. With this, I'm done with this part of general introduction. Is there any question before we move to Brownian motion and very, very physicist way of thinking about it? Any question from this?
Yes. For the part of organoids,
yes.
How do we maintain the internal environment that we have in the body in the petri dish?
This is a very, very good
It
point.
has to
Yes.
interact with other organs and with other
Yes.
components of the body. So how do we maintain that?
So I will make a word of cautious about organoids. At the moment, what has been a big expansion, and now it feels like in many of the lab, and we are contributing to our extent with an international community also, is that growing single organs is quite easy. I mean, it's not very difficult if you learn how to do it. You can do it pretty well. Growing co-culture of organs to have vascularized organ is more difficult. And at the moment, it's going to happen, but we are not yet there. The synthetic embryo I was mentioning, and there are 3 laboratories at the moment who are heavily competing actually and working on it in the last 1 or 2 years actually. As far as I know, they stopped after a week or 2 weeks. They don't want to go above it. But if you think about it, if you follow the right recipe after that, in principle, all the cells have the right genome to eventually, make the right organ at the right place. You see what I mean? That you were mentioning bioprinting. I don't know who was mentioning bioprinting in the way. In self-organization, you don't need bioprinting. You let the system do the job. So if a stem cell changes its identity to eventually at some point make a vascularized tissue, it should, and it would. So the 2 aspects are coexisting together, and as far as I know, when I'm discussing with people from the field, they don't know which one would be the most...
Even if that is designed, but how functional it would be to introduce it inside the body. That's also
So for regenerative medicine, you mean? Yes. I see what you mean. Yes. So it has started to be already applied for some of the examples where in fact it is functioning for some of the organs. Basically, you take a cell, you de-differentiate it, and then you let it follow a path with a target on a given organ, and then it has been started. I want to double check that I don't say something wrong. I think it was for muscles that this has been started. So basically, it can work. And the international effort at the moment is to try to make some medicine, curing medicine, because you imagine the consequences. If somebody lacks a pancreas, you could imagine taking the cell from this patient, de-differentiate the cell to make them be back stem cell, and then re-differentiate them to eventually make them back pancreas. And then there will be a functional pancreas without any need for a donor, without any need for grafts, and without the risk of having a rejection from the body, because they need to be from the same body. It is not science fiction anymore. It's something that can be done, but it's research. It's not yet commonly done in the laboratory. So far, so good? Right. Okay, good. So, maybe one last question before we go to Brownian motion. What is the length of human DNA?
2 meters.
2 meters? Okay, what is the size of the nucleus?
Size of the nucleus should be 1 micron, less than 1 micron.
1 micron. So if you have some problem with having in mind this scale, think about, you're preparing a pullover, you imagine the wool actually, which is surrounded, and you have 2 meters of wool, right? So 2 meters of a DNA. What is the thickness of DNA? What do you say? 2 nanometer. Yeah, I don't know if it's what you said, but it's 2 nanometer. So you have a 2 meter long polymer, right? 2 nanometer in thickness, and you have to put it into 1 micrometer. What's going to happen? What do you say? Folding, yes. What else? I mean, did you ever play with? If you thread, did you ever make a pullover, right? What's happening when you make a pullover? Your mother, yeah, or basically, yeah, I'm sure, Amjay, you've experienced this. What's happening if you don't do anything on purpose? Inevitably, that will get you nervous. It will entangle. Worse than that. I mean, I'm sure you experienced that. I experienced it, so it just, yeah. Knots will form. Inevitably, knots will form. If you take a thread that is extremely long and then you try to sew, eventually knots will fold. So think about the DNA inside the genome. You have 2 meters in 1 micrometer cube space, actually. And eventually you will have some knots. And if you think about it, at each division, because as we said, we come from a stem cell and eventually you proliferate and so on, inevitably you will have some accidents, you will have some knots that you will now to change, etc. And we come back to the point of how can it be working. It is incredible. If you think about first principle from polymer physics, it should fail, basically. And if we have the DNA in our body, which is the very same in our genome for our cell in our body, it means that there has to be some correction mechanism. There has to be some, it's very strange that it can be working because intrinsically this is an issue. And we come back to the same problem of how do you change scale because you will have issues. You know that if there is an error in a sequence or in a base, this is going to be lethal in some of the cases. So come back to the problem of how can it be working. It's okay? So I'll add a level of complexity, which is going to be now the first part, which is going to be about scaling arguments. Anyone is familiar with scaling arguments already or not? You've seen that? Yes? No? Scaling arguments. In scaling arguments, you consider objects, and you don't want to have to write the equation. You don't want to go into the detail of what's going on, but you want to get the right phenomena of what's going to happen. So in a scalar argument, you just think about parameter and you try to think about the right scales. That's essentially the philosophy behind it. With the scaling argument, you can imagine the distance between Earth and the Moon, or you can imagine all kinds of phenomena which are, back on the envelope. And I would like to drive you slowly through the scaling argument at the nanometer scale. Eventually, this could work also at the micrometer scale. All the arguments that I'm going to use, actually, will be essentially for inert matter. What is the difference between inert matter and living matter? It doesn't react? Okay, so it means that any chemistry is going to be active matter. You would say so. Okay, interesting. So basically, any chemical reaction would be an active matter. Fair enough. Let's think about it during the whole week. It's a nice hypothesis. But at the moment, I'm going to bet inert matter. We're going to make it active tomorrow, essentially. But essentially, it would be inert matter here. So as a physicist, I'm going to make a reasoning which is extremely simplified. I'm just going to consider the scale, and the scale that is meaningful, as I was trying to highlight before, is the scale that is here. It's a nanometer scale. So I'm going to take a colloid that is the size 1 nanometer. This is a colloid. What is the energy of a colloid of 1 nanometer in size? Can you give me the energy that a colloid would have? Imagine I give you a bead, and could you tell me, what would you see under the microscope if I give you a bead of that type? Brownian motion, right? Okay, so fair enough, it will have a Brownian motion. What does it look like if you would do this with your hand? It will be this type of motion, okay? KT is what? So KT is the energy. So this is an energy, and the energy scale is KBT. Can you give me the force associated to this energy? So basically it means, for those of you who are not familiar with this, that if I take a particle of size A, I'm going to, instead of taking 1 nanometer, I'm going to say that the diameter is A. I have an energy, I have a size A, I can deduce a force. What is the force associated to the Brownian motion? What do you say? How do you know that? You work on colloid, but could you have a way to find it back? Is there a way for you to, what you said, I will say it loud so that everybody hears, you said that basically the force associated to a colloid moving around is a piconewton. Fair enough. Is there a way, through scaling argument, which is basically the point that I'm going to make here, is there a way to find this piconewton force? Yes, loud.
Sir, force is generally mass into the motion.
Yes?
So mass multiplied acceleration, that is here, we can call it a motion.
Okay, this is exactly what I'm going to challenge today.
Yeah.
It's good that you said it. So basically, you want to go through Newton's law,
Yes.
and you want to find it back.
Exactly.
Okay, right. So we'll go through that, and we'll see that it's going to be a problem. But it's non-intuitive, and that's at the core of what we need to do this afternoon and tomorrow. So it's good. So there is, yeah. So I propose another way to go. Again, with scaling argument, you don't care about the details of the equation. You just care about the units. So what is a force? Loud. Yes. You said something, but I could not hear. Yes, mass acceleration. Imagine that I forbid you that you use mass acceleration, right? Oops. Imagine it's a no, right? How would you do without mass acceleration?
So what is the question exactly again?
Can you? What is the unit of a force? Newton.
Yes.
Newton is what? Fair enough. How do you relate force with an energy?
So you're going back to the units MLT.
I just want you. Right, so basically I would like, and this is what I'm going to ask at the moment, is if you take a force, if I have an energy, and if I have a scale, dimensional scale, spatial scale, F, E, A, can I have a relation between all of them that is going to allow me to have the right unit for the force? What do you say? All right, so F is going to be given by E over the size. Okay? So basically, it's the ratio of the energy over the scale of the object. And so this ratio, for those of you who are not familiar with this, is essentially, if you want to think about it, it's dimension analysis. You have some units, and when you make the dimension analysis, this is a combination of parameters that can make sense. So exactly to the point of you were telling me, Katie, for those of you who are not familiar with KT, this is the Boltzmann constant, this is the temperature in degrees Kelvin, and the multiplication of both is giving the energy. So it's a very deep and very it's not super easy to think about it, but basically KT is the energy of any object of that size. And I'm ignoring the molecular detail. I just want to care about the size. So F is the order of E over KT. If I plug in number now, the Boltzmann constant is equal to what? 10 to the minus 23. So it's 10 to the minus 23. The temperature is 100. So 3 10 to the 100. 10 to the 2. And A, I'm taking a particle of size
1 micron.
So 1 micron would be a cell. So I would be here. 1 nanometer. Right. So I propose to take it because you will see that the result is interesting. So basically, let's say 1 nanometer. So it's 10 to the minus 9. So this is going to be 10 to the minus 23 over 10 to the minus 9. I ended up with what? 10 to the minus 12, and you have your piconewton. Right. It is amazing that somehow you got the right number, but in 2 minutes, with the scaling argument, you find it back. So basically, it means that whatever I do, I will have, when I see a particle moving, and this afternoon we'll hopefully see some particles moving live, right? I'll show you a movie, but just at the end for fun. But just before that, try to think about it, right? If you have a particle like this of nanometer in scale, it will have a force that is 3 piconewton without me doing anything. I just observe it. I just see it moving like this. So I know that I was a bit imposing the argument that I don't want a mass and an acceleration. I'll try to detail in 20 minutes why. And there is a reason for that. But at the moment, just bear with me with the fact that without going into much detail, if I take a molecule just because there is a temperature, it's going to give a force of the order of piconewton. Now I want to move forward, and I would like also to give it another gist, actually. That will be the diffusion constant. Anyone knows what the diffusion constant is? Fair enough. For those of you who never saw that, so that we are all on the same page. If I take a colloid, if I take a particle, the particle will be jiggling around like this. How can I measure its motion? If you want to measure a motion, what would you use? What do you say? Mid-square displacement, this you're already familiar with. For those of you who are not familiar with, how would you do that somehow? You see the problem? That what is the readout that you would be using? If you want to quantify a motion, what do you do usually? What do you use? At the back. If you want to measure a motion, what do you use as a readout or as a parameter? Displacement, but displacement you lack time. So if you want to go to the center of Delhi to the other side of Delhi, what you will care about is, what do you say? Different point, but what you care about to arrive on time is? Yes, but if you want to arrive on time to your meeting, you need a different point, and you need also the information of speed, right? Now, if you have a motion like this, which is erratic, is the speed meaningful or not? It is? Well, if... All right, but imagine you want to go from center of Delhi to the outside of Delhi, and then you go backward, forward, north, south, in every direction. The probability that you will reach the point of your meeting on time is pretty low, right? So basically, you are in a diffusive process, and you need a quantity to measure this. And the point that I'm going to highlight with you is that then you need to think about something that is random, and the quantity that is going to capture it, and you got it already before, is the diffusion constant. And I would like that we go slowly through other scaling argument with the diffusion constant. So for this, I'm going to use the Einstein relation. Anyone has seen it before? Einstein relation, fluctuation, dissipation theorem. For the most, never mind, these are words, you've seen it in the past. So basically, it tells you something very profound, that the diffusion constant is equal to kT over 6 pi eta r eta A. You can derive it. You can show that it is correct. So I would like you to try to get a sense of what's going on here. I take a particle. This is the colloid, right? The colloid is here. It receives some thermal shock by the particle, which are giving some kick everywhere. So if I am the particles, I'm receiving some shock like this, in every direction. I cannot go z because gravity is making me going down, but you can imagine that it would be in 3 dimensions. This is encoded by which term in the diffusion? What is conveying here the fact that I'm moving around everywhere? It's okay with everyone? What is the energy of a particle of 1 nanometer? Katie, what is the energy of a particle of 1 micrometer? What is the energy? No. Katie, what is the energy of a particle of 500 nanometer? Thank you, Katie. So basically, at the end of this lecture, I would like you to remember only one thing is Katie, right? Katie is going to be the energy of all the objects we are talking about, which are here, when they are inert, basically. So I'm having Katie. Katie is here, and I need to define the terms that are below. So here I have A, which is the size of the colloid. Here I have the viscosity. Did anyone see this term already before? I mean, there won't be hundreds of equations this week. For those of you who are the less formal, no worry, but at least these ones, I need them. So what is the, what is the term that is in the denominator? It's a drag constant. So both of you, I think you're already familiar with this. For those of you who are not familiar with this, what is viscosity? Yes? Yes? So density and viscosity are different concepts. What do you feel when something is viscous? What do you say? Friction. Yeah. I mean, there is something associated with friction, which is when I'm trying to move something, when I'm trying to move a colloid, if I am in a viscous fluid, I will have a hard time, moving it. I mean, there will be something that will be difficult in the motion itself. This is totally right. Yeah. So here, this term is the product of the viscosity times the size, right? And anyone has seen that before or not? If you get an intuition of that, if you play with honey, if you put your spoon in the honey, what's going to happen? There will be a viscous component, meaning that the motion will be impeded by the viscosity. So try to think about the meaning of this term. Here I have the energy. This is the energy of the colloid. Below here, I have the viscosity, I have the friction. So I have a ratio between me moving all the time and the friction surrounding me. What's going on here?
It will just tell whether the particle will diffuse in the system or will be spacely depending on the ratio what we get. Like if the
Yes.
numerator is greater than the denominator, the particle will move.
You're absolutely right. It's perfect summary. This is a ratio between both. But something that you cannot escape if you are an inert particle is kT. kT is a temperature. So basically, if I have a particle that is embedded inside a bath at a temperature T, I am bound to be having this energy, kT. That's what you said before, and I totally agree with you. So basically, the energy of this object is kT, whatever I do. I cannot escape it, unless I reduce the temperature to zero absolute, right? But otherwise, I'm moving all the time, and kT is a given, so I cannot escape it. So I have this ratio that is here. So orders of magnitude are interesting. I'm talking about living matter. So basically, I'm going to say that living matter is in which environment? I mean, basically, life, usually, you need what for life? Water, right? So basically, I'm going to take the viscosity of water. And the viscosity of water is equal to? Yes, unit. It's good. It's very, very good. Thank you so much. This is 10 to the minus 3 Pascal second. So now we have everything, right? I'm going to write it here. So D is equal to 3 10 to the 2, 10 to the minus 21 over 2 times 10, 10 to the minus 9, because I said I would take a particle of 1 nanometer in size, can multiply. Sorry, first you need to add 10 to the minus 3 and then 10 to the minus 9. In scaling argument, we are not super nervous about a factor of 2 or a factor of 5. Scaling argument is really back on the envelope evaluation of what's going on here. So here I have 10 to the minus 19. And here I have 10 to the minus 6. I end up 10 to the minus 13 meters squared per second. Everyone agrees? I didn't make any mistake? It's okay? Okay. So if it is meters squared per second, I'm going to convert it into a unit that is a bit more pleasant. It's going to mean that I have about, there is a mistake somewhere. It's okay?
So there's a 10.
So it's, oh no, I forgot something. It's 3, 10 to the minus 23 here. Yes, it was 10 to the minus 23, so it's minus 21. 10 to the minus 15 meters squared per second. 3, 10 to the 100, 10 to the minus 23, 10 to the minus 21. I'm going to check on my note, but I think it's correct. Yes. So if you convert that, you end up with a diffusion constant that is going to be equal about 5 micrometers squared per millisecond. I would like you to try to think about this number. This is about, right? I mean, this is really an order of magnitude. What does it mean if you have a particle of size 1 nanometer that is going to move 5 micrometers squared in a millisecond? Try to put it in perspective. I have an object that is nanometer in size, so I have a DNA that is in the middle of the room, and somehow it's going to be moving everywhere. So imagine the wall of this room is essentially a cell, and I have a small protein that is in the middle of the cytoplasm or in the middle of the nucleus. It means that in 1 millisecond, it will have gone through a surface that is 5 micrometers squared. It is gigantic. It means that somehow everything is moving constantly. If each time I have a protein, an RNA, or a DNA, in addition to the problem I was mentioning before about having a huge genome that need to pack at some moment, if you don't do anything, a small object will be going everywhere. So again, the metaphor is an object in the center of the room that a millisecond after will be at the end of the room. And this is a given. It's something that a cell or living matter cannot escape. So it's somehow embedded profoundly inside the system itself. So this relation, which is essentially illustrating the diffusion constant, is going to be also a key aspect. Let me summarize where we stand. With scaling arguments, you can somehow have gut feeling about very, deep, profound understanding, or at least intuition, about what's going on into the information scale, which is at the nanometer scale. First of all, you have an order of magnitude of force, piconewton. You have an order of magnitude of the diffusion constant, and for this you don't need to make a sophisticated experiment. I mean, essentially the scaling argument is here to help you and to drive you into the calculation. And you end up with an intuition of what could be happening inside, inert matter or living matter if you don't add something that will eventually add tomorrow. Now, the last question I would like to ask you is, anyone knows what inertia is? Inertia, yes. Yes, inertia. Yes, so what is a typical example of inertia?
You are on the moving bus and
That's right, that's right. So I'm going to make an experiment that is very, very difficult. So you ready? So I have this. 1, 2, 3. Sorry, I'm sorry, I missed it. It's my fault. But somehow this could work. Thank you. This could work because when I was having my stuff here, when I was throwing it, after it has left my hand, it was reaching you. This is, back on the envelope inertia, right? It's sort of fun. I mean, with this you can play cricket, right? You can, it's really like without inertia, no way you could eventually have this type of sport. You couldn't make bicycle, etc. and so on. So the last question I want to ask on this actually is it a world with inertia or with no inertia? I'm repeating my question, is that whenever I have a particle of 1 nanometer in scale or 1 micrometer in scale, it's moving all the time, right? It's kT. So basically, I'm moving this, I'm doing this like this. Is it with inertia or without inertia? And I'm happy to, those who know, those who don't know, try to think about it, because otherwise, I mean, you need to have a question before having an answer. Otherwise, it's, Yes, with inertia, right. So there is inertia. Everyone who says there is inertia? Raise your hand. Right, there is inertia. Good. Who says there is no inertia? Okay, minimal, right. So basically, massive, there is no inertia. Okay, good. Why?
opposed motion.
Yes.
And this motion is happening at such a short time scale.
Yes.
It seems like if the tendency to oppose the motion would be there, it would not probably be able to cover as much distance. It would be more stationary than moving.
Okay. Okay, I understand the argument. And those who say there is inertia? Why?
Inertia does not, the exact definition of inertia is not the tendency to oppose motion, but it is the tendency to resist change. If it stays in motion, it wants to stay in motion. If it stays in resting, it wants to stay in rest.
Okay, I see your point. I follow your argument. You see that in the way you formulate, there is the word want. I'm talking about the particle. I'm just provoking you, as you understand. But somehow I agree with your definition, but if I have a particle, the particle does not want anything. It's a colloid, right? So the colloid is going to move somewhere, and at some moment it's going to be pushed somewhere else, and then it's going to push somewhere else. But I think massively, and I was the same like you, somehow, and still somehow, is a way of saying that, well, it's going to be with inertia. So with inertia or without inertia? It's important, because it means that if I have a particle inside living matter or in inert matter, well, eventually this particle will be having inertia, meaning that I don't need to provide energy to move. I mean, when you are on your bicycle and you stop, I miss the word in English, but you see what I mean. I mean, you stop making some effort. What do you say? Yeah, you stop paddling, eventually you will continue, and actually you will benefit from the energy you had before, like what I was doing before. So it's particularly important to know if you have a polymerase, a DNA polymerase that is allowing basically to have an RNA, if it doesn't need energy, if there is no inertia, it will continue to move, and then you continue without any source of energy. So the fact of knowing whether or not you are in a world with inertia or without inertia is not a minor detail. It is essential. Like, you need to know that. If you have a growing organism, an organoid, which is changing its shape, is it changing its shape because there was some velocity before, and it continues because it just continues what it did before? So again, it's not a minor one, it's a central one. How can you know whether or not there is inertia or there is no inertia? Difficult. So for this, scaling argument, that's why I was willing that we go through scaling arguments first. So I'm going to go one scaling argument through a number that is called the Reynolds number.
Sir,
Yes.
I got two questions.
Sure.
So the first one is about the diffusion constants. I have not seen this equation. I mean, I forgot this equation if I've seen in the past. But there is no addition of the mass here, so diffusion does not depend on the mass. That's exactly the point that I'm going to. Okay, one. And second one is the units of this diffusion constants is meter per second,
Yes.
meter squared per second.
Yes.
There are also the units for acceleration.
No.
No. Meter per second squared.
Yeah.
Okay.
Yeah.
So that's fine.
That's basically both questions are extremely right to the point. Is that you feel like somehow all our intuition, because we live in a world which is the world of bicycle and transport and so on and soccer and you name it, that somehow we have all our mind, all our brain is towards this, and so mass is entering into the game. Everything that you said, I mean, I totally agree with everything you said. I hope that by 10 minutes you'll be convinced.
Okay.
So the Reynolds number is a number that is extremely useful because it's a number that has no unit, so it's easy to manipulate. And it's basically, maybe I will move it if it's okay. It's the ratio of the inertial force over the viscous force. So since it's a ratio of force over force, it has no unit. So it's very convenient. Why it's very convenient? Because then, if the Reynolds number is very large compared to 1, everything is dominated by inertia. If the Reynolds number is very small compared to 1, everything is dominated by viscosity. So basically, it's a way to fairly estimate in which case you are you in presence of inertia or are you in the absence of inertia? So I'll give you the expression actually of the Reynolds number. And again, it's impossible to get it with intuition. You need to write the number. You need to write essentially what the number gives. And so if you write it, it's rho a nu over eta. So A, it is the diameter of the colloid. This, I mentioned it before. Rho, and that's why you were talking about density. I don't know your name yet, but you were talking about density. So basically, do you see that the density is another one? It's another physical parameter. And I agree that both of them meet, but they are really having their own autonomy. So this is the density. This is the viscosity. And V is the velocity. If you compute the number, if you take it essentially for water, you end up, I will let you make the calculation, to 10 to the minus 4. 10 to the minus 4. I'm taking again my colloid, which is A of the size of 1 nanometer. So it means that if I take a colloid of size 1 nanometer, I have a Reynolds number of 10 to the minus 4. As a consequence, all the life at low Reynolds number, which is the title of a famous article by Purcell, is with inertia or without inertia? With inertia? With or without? So I'm going to write it so that basically there is no ambiguity. If the Reynolds number is large compared to 1, basically it means that the force is viscous inertial force is larger than the viscous forces. So then I have a life which is with inertia. If Reynolds number is very small compared to 1, I have a life without inertia. So if I have a Reynolds number of 10 to the minus 4, you're convinced? Okay. Sure? So basically, now I would like to raise your hand. The life at nanometer scale, there is or there is no inertia? Who says there is inertia? Who says there is no inertia? Okay, good. Yeah. You need to speak loud.
I'm not understanding. If you're saying that if Reynolds number is smaller than 1,
Yes.
the inertia should not be there.
Yes.
But how should it not be there?
How could not that be there? That's exactly the problem, and this is the whole point of starting the lecture by this. Is by starting by saying there is a problem, that somehow you have the Reynolds number, this expression is correct somehow. If you write the inertial force over the viscous force, this is basically a result that it's pure math. So I mean, you cannot do anything. That's what it is. And now, if you plug in number, you end up with 10 to the minus 4. So basically, it's just screaming that there is no inertia, even though intuitively, massively, you and me also, when I was at your stage, I would have said there is inertia. There is no. There is no. Forbidden. There is no inertia. So everything that you said about mass, I mean, this is normal because we are used in a world with inertia. That's basically where we live in. But the point that I'm starting with is that the meaningful information by which I was starting on the top here, left actually, is the fact of saying that no, there is no inertia. So yes.
So how did you get to that number?
I'm sorry.
So how did you get to that number?
You plug in number. You take A, the size, viscosity of water, you take the density of the water, and the velocity, you take the force I was giving to you before, And basically, with the velocity, you end up with another expression. You have 10 to the minus 4. I can, if you wish, we can do later on the calculation and the derivation. It's the same type of calculation that I was deriving here, but I take it for water, essentially.
But the density is also dependent on the volume. And volume is
The density is the density of water. So basically, that is a constant. That's why I'm saying that somehow everything that I'm taking here is constant. So I'm plugging in the number of the density of water, the viscosity of water, the size of the colloid, the velocity that the colloid has, because it has a kT that is around it. And then if you plug in number, you end up with a 10 to the minus 4. It is somehow a given that is coming out of the calculation. And so, again, I think this is the paper I wanted to treat on Friday, but we'll see how the discussion goes. This actually has been amazingly described by a historical paper, really historical, which is life at low Reynolds number by Purcell. That somehow he's saying that whenever you think about life, whenever you think about living matter, there is an intrinsic problem is related to the fact that there is no inertia whatsoever. You can turn the problem everywhere at the scale of the nanometer, which is again the magical information scale. At the scale of the micrometer, at the 1 micrometer, there won't be inertia, and never, ever. It's forbidden. So then you need to invent or to think about the system in a very different way. Yes?
By saying there is no inertia, we are considering those particles as massless at that scale. Is that your...
They have a mass. There is a density, so there is a mass. I mean, the particle has a mass. You're totally right. It has a mass, but somehow it's negligible. The point is that it's not anymore the important point. It is a problem, right? So you say it's a problem, yeah. So maybe I will go through another side so that you feel a bit why it is true, actually, is that if you write now rigorously the equation, which is you're going through the Langevin equation, for those of you who've seen them most probably, probably you are used to be going through the rigorous math where you will have, in one dimension, there's going to be a term associated to the velocity. What do you say? So it's going to be, I'm going to write it alpha. I'm going essentially into 1D. That is the fact of doing the very same calculation as before, and this is essentially Newton's law, right? You have mass times the acceleration, which is going to be equal to the sum of the forces. If you plug in number, which is essentially what the Reynolds number is doing, essentially you end up to the conclusion that is also disturbing for everyone, is that this is negligible. It is only because you make the calculation, right? It's because you plug in number. And when you plug in number, you see that somehow it's peanuts, it doesn't count. So the consequence, and this is the last point I want to share and then try to brainstorm about the consequences about everything we talked about, is that in this type of physics, whenever you have a motion, you have the forces that are compensated by the velocity. So each time there will be a velocity, it doesn't mean that there was inertia before. It means that there is a new force being applied. If I see a vesicle moving like this inside the cell, or if I see a molecular motor moving DNA polymerase, RNA polymerase, F1 ATPase, you name it, all these motors, somehow it means that if there is a velocity, it means that there is a force being applied and there is a need for an energy. So I'm going a bit fast here, but I will repeat that again tomorrow and probably the day after. It's somehow, at this scale, you need to reset everything you've been used to. I will summarize to give you another intuition to see why I'm going in this direction. Imagine if you would play soccer. Why can you play soccer? You can play soccer or cricket. I think here cricket is, yeah. In France, soccer is the one, right? So, but, okay, so if you
in French, soccer and cricket are synonymous.
what do you say?
In French,
Soccer? Soccer is not football.
Football,
Yeah, it's soccer. Yeah. If you wish, we can play soccer. I'm pretty poor player, but we can. Yeah. So, but yeah, it's like this, and yeah. But basically, in soccer, you can play soccer because somehow the ball is going to get out. And cricket, you will teach me cricket if you wish. But it's basically you see what I mean, right? So, what would be the equivalent of low Reynolds number cricket or low Reynolds number soccer? What do you say? Cricket, low Reynolds number cricket. Let's dream about it. Yeah. In a pool of water, that's essentially like this. Imagine a swimming pool in which there would be on the ground all the marking of the cricket, and then you would have the opportunity to play cricket. Yes, you say no, no way. It could be interesting, because then it would mean that what? What won't you be able to do that you are using in playing cricket?
inside a pool.
Inside the pool, that's right.
So we cannot be able to, swim though. It's bad.
It's bad.
Yes.
Right? It's different. I'm not saying it's bad. It means that for eventually having something interesting to do, you need to do what? You need systematically to apply force.
Yes.
Basically, each time you are I missed the word in English, I'm sorry. In cricket, you are hitting is the right one?
Yes.
So hitting the ball? So if you hit the ball, but if you hit the ball, it doesn't change somehow. You need to constantly, there would be a need of contact, because then the person opposing you, they will need to come in front of you and to challenge the ball and to take it. That's essentially the consequence. So it would be cricket at low Reynolds number would be cricket inside a swimming pool. Let's think about it. Right. Now, I'm coming back to the cell now. If you take a particle, if you take a colloidal DNA, it has this problem, but in addition, it has an energy which is kT. So it's a complicated one, because it means that if I am DNA, I'm moving all the time like this because I have shock from the particles around me, which are giving some shock like this, but there is no inertia. So it's extremely difficult to have a good intuition out of it. It means that whenever, and we're going to see this afternoon, tomorrow, some real data where we will see some colloid moving and some molecular motors moving, etc. It means that they're going to be kT. Whenever there is kT where the energy is purely inert matter, I'm moving all the time, but it stops immediately. So there is no inertia whatsoever. It's okay? And this is what I would like you to remember, is that all the life that we're going to talk about is going to be at low Reynolds number, and this is an essential property of inert matter, whatever it is. Questions? It's okay?
So essentially you are saying that by considering without inertia of that particles, there is no type of collision. They are just colliding and changing their speeds.
I'm saying that if I see a particle moving like this, it has energy which is kT, right? But it has no inertia.
It collides and changes its speed.
It has collision from the molecule of the solvent that are giving shocks. So basically that's going to promote the motion, but the statistical distribution of the shocks on the particle is going to make it go somewhere else at some point. And the inertial component of its motion is peanuts. Most of the motion is piloted by shock of the particles that are going to make it move. For those of you who are making FCS, I think some of you are doing FCS, I heard yesterday evening. So basically FCS, or if you see a protein folding or protein fluctuation, or you name it somehow, it's essentially this that we are talking about at the moment. It's basically kT There is no energy in everything that I talk about. I am purely passive today. I'll be active tomorrow. But in a passive motion, essentially, I receive that, and this type of motion is totally disconnected to any inertia. Cricket in swimming pool. But I need to add that it's not exactly cricket in swimming pool. It will be cricket in swimming pool plus, is it called the ball for the cricket? Is the right word? Okay. So the ball, in addition, would have a spontaneous random motion. That would be fun, because then the ball would be, randomly escaping the spot, and then people would need to chase to be able to have it. But this is the world that we are talking about. Yes.
So, in order to understand, because this is very, very counterintuitive at that stage.
This is totally counterintuitive. This is the punchline I wanted to reach. It's done something. It's exactly counterintuition. So basically, your intuition, my intuition is wrong.
Yeah,
yes.
So in a cell, for example, particles are anyway moving.
Yes.
Yes. So it seems that there is inertia, but there is not, as you
That's right.
explained.
Yes.
But they're anyway moving. So what is the initial force is driving them to move?
This is the question.
Okay.
Right. This is the question. Totally agree. At the moment, I inert. Everything that I said here was inert. Right. There is no inertial. There is no external energy. I take a colloid. I take a DNA. It's moving all the time. I haven't put energy. Tomorrow we'll put energy tomorrow morning. But we'll try. but basically here, I'm just taking KT. I'm just saying the temperature is, temperature, we could argue, is energy, right? And this I was saying since the beginning. So you could say, well, energy could do the job. Well, we'll see tomorrow that no, it's not doing the job. Okay. So actually, there are 2 or 3 last points I would like to do before showing you a movie. I was escaping PPT and movie because I think it's easier for board. You tell me if it's okay, but I think board, it's easier to interact with the board. So think about the consequences of KT and diffusion, right? I'm repeating, but I think it's really key to have it in mind. You remember the DNA 2 meters long in 1 micrometer in size? So it's like a thread of wool that is not everywhere. Add to that diffusion. It's a mess.
Yeah. It is trying to hold or
yes.
organize, but the inertia, the motion, random motion will.
It might, depends on orders of magnitude. You need to check if the viscosity will match also. But if you don't do anything special, somehow you have your DNA that you think about inside the nucleus that is going to be moving everywhere all the time. Is it right? Is it wrong? We'll see. But somehow it's going to be very, very dramatic. Another example that is going to be important for the lecture of Thursday. We'll talk about one circuit inside the cell, which is the repressilator. Anyone has seen the repressilator already or not? Circuit which is called the repressilator. So this will be for Thursday. It's a minimal signaling network with, you can write the equation, you can predict, you can compute them, you can make simulation of it, and it's extremely powerful to understand some signaling network inside the cell. I'm tackling this. Imagine you're going to have a gene and a transcription factor that should find the proper spot in the genome. What the transcription is going to do inside the nucleus? It's going to?
Sir, what is the question?
If you take a transcription factor, basically if you take an essential protein that eventually, a complex of protein that is going to find its target on the genome to eventually express a gene. You have the nucleus, right? You have a transcription factor that is here. To find its target, what is it going to do first? It will diffuse, right? So it will diffuse everywhere. What is the probability of a transcription factor to find its target of a 2 meter long DNA inside the nucleus of 1 micrometer cube, bearing in mind that the size of the transcription factor is few nanometer?
Yeah.
It's very low. So you come back to the point that needs to happen. So basically, even expression of genes, which are essential, is going to be prone to noise. It's going to happen or it won't happen. You will have a low number. So basically, you come back to the fact that because of this diffusion, you end up with some biological consequences that are going to be extremely important. Right? I'll give you a last example. What is the most efficient way to go from Delhi to Bangalore? Take a flight. Okay, the flight will go in which way from Delhi to Bangalore? Yes. Taking some detour or straight? Straight. Okay, right. I'm saying my question is stupid, and I know that. Now imagine you have a part of a cell that is broken. What is the most efficient way to repair the point of the cell? Go straight, right? You have a machinery that is going to fix the point that is broken, and then you will fix it. If you don't do anything special, what will happen? Everything will be dominated by what? Diffusion. So it means that if I take the metaphor of, going from Delhi to Bangalore, it will mean that you will travel throughout India, and eventually one day you will reach Bangalore. It's exactly the same problem, that somehow, because all this world is totally dominated by diffusion, you have this problem that is showing up, which is always present.
Sir,
Yes.
so what is at stake? Because to cover any such distance,
Yes.
let's say inside a cell,
Yes.
so a particle would want to,
The particle does not. I'm sorry to interrupt you. The same way it doesn't want. It just the particle is KT,
so
right?
the design of
Yes.
a cell
Yes.
is such that we intuitively think that a particle should is the right word or not? Particle should travel at the site of injury faster, right?
I didn't say speed. I talked about randomness or non-randomness. I just say I'm comparing a situation where this is KT, right? And I'm comparing this with the situation by this one. This is all I'm comparing. I didn't talk about tangent velocity. Yes.
So what I'm saying is, but anyway, this motion, the random motion takes time, right? And the straight
Yes.
path does not take that much of time.
The-
We are compromising the time. Then what is at stake? Why the diffusion is important, but the traveling in a straight line is not that important?
Actually, I didn't say that diffusion is important and going directly is important. I'm saying that there are some contexts in which diffusion will be important. It's a built-in property. And I'm saying that we'll see them this week, some of them, is that there is some context where directed motion is essential. If you want to reach Bangalore on time, you'd rather go straight, right? If there is a need to fix urgently a part of a cell, it's rather be efficient and go directly to the point, right? So there is some point where diffusion will be nice, because with diffusion, if you wish, you will encourage randomness. You will have a random event that will eventually happen. If you are in a directed motion, well, you will benefit from other feature, which essentially precision. You will go exactly on the right spot. Okay. So to conclude, it's okay. Other questions? I know online there might be some questions, but I don't see them. So basically, it's okay. So I want to conclude by two movies, and then we'll stop. Maybe one movie, and then I'll just summarize. Let's say two movies, and we'll come back to this movie tomorrow. So the movie number one is going to be a movie that we're going to see this afternoon. It's a particle of 2 micrometer in size.
That black.
This one? So probably the light is going to start in a second. Can you see can you see the signal? No. The light is not, the light is starting, yeah. Yes. Right. Maybe if you can see. So this is a particle of 2 micrometer, right? That we keep talking about since 2 hours now. So basically, it's going to give this. What do you see? Yes. Why is it going out of focus? Yeah, KT, right, KT. So basically it's moving X, Y, it's also moving Z. So basically this is this motion that, who hadn't seen Brownian motion before? Who had seen Brownian motion before? No one. Great. This is the whole point, is that it is so nice because somehow it's temperature, it's everywhere, you cannot escape it. It's like molecular scale and the micrometer scale is totally embedded into that. So basically this is what we will try to see this afternoon with the microscopes, right? And this is a 2 micrometer particle. I'm showing it one last time. And it's so easy to do, right? You just put a particle inside the microscope and you let it go. And now what I would like to show, and you know, we'll see this is a gigantic gap, but I think it's a gap that is interesting to do actually. It is, you know what, maybe, okay, I have prepared it, I would like to show it to you to show you as a difference actually, one that actually I had prepared it. I hope I'm going to find it quick, otherwise I'll show it to you tomorrow morning. I don't see it right here. Basically, I think, yeah, at the moment I was totally inert matter. I just would like to wrap up and I'll show you the movie of directed motion tomorrow. Basically, at the moment today, we are essentially inert. I have 3 more minutes and I would like to wrap up to say everything that I've been presenting today. So, I was starting with a general introduction about the spatial scale of living matter. This is essentially a spatial scale with very minimal information. I mean, of course, I invite you to read introductory books about the basics of the cell, the compartment of the cell, the molecular component of the cell. But in terms of general vision, it's one way to present it, just bearing in mind the scale. I was saying that the meaningful information, which is conveying information, is the nanometer scale. This is throughout living matter or living systems. The scale of the cell is micrometer, 10 micrometer. Bacteria, prokaryotic, eukaryotic. When they start proliferating, eventually changing their state, you end up with a tissue that eventually leads to an organ, that eventually leads to an organism. And one of the questions of biological physics is in terms of signaling network or in terms of changing scale, how do you go through self-organization? That was the part number 1 that I was starting the lecture by. And then I was saying, if you ignore the chemistry, just for the sake of reasoning, could you again come to simple arguments to try to understand what is the physics associated with the phenomenon? And now I was essentially in a passive system. I was also using the word inert, but passive or inert is synonymous. Somehow I'm just looking at the system as they are. And I was giving scaling arguments, and I went through several of them without formalism, just taking scales and units. A particle has an energy kT. I can derive a force. The force is few piconewton without me doing anything. There are some instruments to measure this, such as optical tweezer or AFM or magnetic tweezer. This force can be measured experimentally in a quite exquisite manner and resolved in time. Then I was going to the diffusion constant and the Einstein relation, which allows me to have the ratio between the energy of a system, which is resisted, and now it comes back to one of the points we were saying before, by the viscosity. You feel it, like here, the contradiction, which is counterintuitive, is here. It is in the diffusion. You are comparing an energy of a system jiggling around by the viscosity that is surrounding it. So it is encoding very deep physics, very, very deep physics and counterintuitive physics. It is here. And you see that it depends on very few parameters, viscosity, temperature, and size. It seems the counterintuition that it should give inertia, and the Reynolds number that we computed here shows that, in fact, inertia is essentially negligible. So we need to think about living matter in a world which has no inertia, with all the problem it comes with, diffusion meaning noise, And at the same time, no inertia, meaning that systematically I need a force if I'm going to see a velocity in a phenomenon. So today, tomorrow, and on Wednesday, we'll be playing around this concept, trying to make this inner matter being a living matter. Bon appétit, as we say in France. See you later.